All right, so kind of a quick review on this alleles. What is an allele? As you can see here in this problem set, it's pretty easy. You have a bunch of chromosomes, 1, 2, 3, 4, 5, etc. And you'll just notice that there are 2 copies of each chromosome, and everybody should know that. You get one set of chromosomes from mom and one set from dad. By this time in this lab, it should be very clear where the chromosomes come from, but I'm still getting questions of like, where is this chromosome coming from? I think I was just asked even by a student a little bit ago, how do I know, what sex is this embryo? They couldn't answer that, and that's really kind of basic, right? So if it's a Y chromosome, if it has a Y chromosome, it's a male. If it has 2 X's, it's not if it has an X, it's a female. No, if it has 2 X's.
Yeah, that's what I said.
Okay, I didn't hear the 2. All right, so as we're going through this, you see that there's 2 alleles available. And why are there 2 alleles available? Because there's 2 chromosome number 1s. And so each chromosome has the same genes on it. So chromosome number 1 has the same genes as the other chromosome number 1. The difference is that the alleles, that there's a different version of the genes where this one is dominant over this one. If you have a big A, that's one that shows up in the gene product. Remember, the gene product's a protein. If they're the same, in this case they're heterozygous. Why are they heterozygous? Because you have one big A, one little a. One version that's dominant, the other is recessive. And these are homozygous dominant. Why? Because they're both dominant. They're both capital in this case. And I hope, I shouldn't have to say this, but I want to be clear. This is not the letter big B. This letter big B just represents that gene. That gene is a number of nucleotide bases. So it's nitrogenous bases. A whole series of them, A, T, G, C, 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 etc. Hundreds, thousands of nucleotides long that define that gene. You should know that by now. We've covered transcription and translation. You should know that that's what this letter B represents. This big letter B means that this gene produces a really good copy that if it's there, it shows up. This little a is a copy of the gene. I hate using the word good, so maybe I should stop using that. It's a version of the gene that if you see the other one, this other one shows up. They're both controlling the same trait. It could be skin color, it could be color of the flower, it could be height, it could be weight, whatever. But this version of the gene, both genes control that trait. But if you have this gene, that version of the trait shows up. If you have this one, you need 2 copies of this, homozygous recessive for that version of the trait to show up. So here's a copy of a homozygous recessive, 2 little c's. So that's what we're talking about when we're saying dominant recessive. When you're doing a Punnett square, someone asked me this morning how to do a Punnett square. I'm glad people are asking me. I'm a little frustrated that by this point you haven't gotten it. You haven't come for tutoring if you haven't gotten it. What you're going to do is you're going to look at a parent, and if a parent has big A, little a, then that's the parent's genotype, and that's what you put here because that's what the parent can give. The parent either gives this chromosome, which means it gives a what? A big A, or it gives this chromosome, which is the little a. This is all that parent can give for that chromosome, one or the other. These genes are all linked because they're all on the same chromosome. And in the lab, now this is one of the lab questions for you. In the lab, they ask you, okay, well, first of all, in the lab, I told you we're not going to worry about crossing over, right? We're not worrying about crossing over. So that's the only way to mix these up, right? Because as it stands now, you're either giving this chromosome or this chromosome. That means you're either giving big A, big B with a little c, or you're giving little a, big B with a little c, right? That's the only 2 options you have. When crossing over occurs, and we've gone over it, there's a whole video on it, I've already posted it, we've gone over it in class, you've done work with it. If you don't understand it, you need to come for tutoring. But crossing over happens, you mix these up, so now instead of just 2 versions, you'll have 4 possible versions in your gametes, right? But in your cells, in every cell of this mom, if this is a mom, then this is the only options mom has. Mom only has this chromosome or this chromosome. That's it. So mom can give a big A or a little a, and dad can give a big A or a little a. Let's say both are heterozygous. So what that means is that either dad gives this chromosome that has the big A or this chromosome that has a little a. All right, that's it. So the possible zygotes, the possible genotypes are these. And that's it. So you have half of them can be heterozygous, 1/4 can be homozygous dominant, 1/4 homozygous recessive. Those are your options. That's what the Punnett square is about. And how do you mix this up? How do you, in this lab, we artificially only gave you 2 options. Artificially, you only had one chromosome or the other. In a lab, in the real world, meiosis increases genetic variation by crossing over. That's going to mix it up. All right, so now Mendel's laws of segregation. Mendel did some experiments. He looked at traits. Remember, he didn't know what the genes were. He had no idea what genes were. He didn't know what DNA was, right? He's a monk in Austria, shucking some peas, making some pea soup, or getting a salad together, whatever he's doing with the peas. And he noticed that they have different shapes. So some plants gave him wrinkled peas, others were round, some were green, some were yellow, some were green pea pods, the other were yellow pea pods. There were some were axial and some were terminal. In other words, some had axial meaning at the axis, right? And others were at the terminal, so they were at the end, right? And some were constricted so that you saw, like, from the pea pod, you see the little peas inside. Others were inflated, so you couldn't tell where the peas were in the pea pod. And he looked at stem length. How long were the stems? Were they short? Or if they were short, he called them dwarves. And if they were long, he just called them tall, right? Taller dwarves. And he just counted them. He counted them. He said, wow. Imagine counting 7,324 wrinkled or, total peas and deciding how many wrinkled, how many round. That's just, to me, this whole effort, it would take a monk to do it, right? Because you have to have that kind of patience. So he made all these counts, and now you're going to go ahead and do it. I want you to do this. I want you to write down how many dominant, how many recessive. You can see the round here, he felt were dominant. Why do you think the round were dominant?
Because it was more.
Because there's more of them. And he said, and so he looked at how many of each, and he made that decision. Now, this is a little misleading because, well, it's not really misleading. If the gene is there for round is going to show up. So you're going to see more round. I don't think that's hard to understand, right? But in a population, that's not necessarily true because we have something called natural selection. So it gets a little more complicated, and that's the next unit after chi-square. We're going to talk about populations and how those things change. All right, so that's what Mendel did. From all this work, he came up with these theories. And he said there's this particle. He didn't know what the particle was. Today we know they're what? What do you inherit? Genes, and the genes are on what? Where are genes? Where are genes? On chromosomes.
I said that the first time.
So on chromosomes, I didn't hear you. So they're on chromosomes. So those chromosomes are what Mendel called particles. So he knew you had particles that you inherited. Those particles that you inherit today we call chromosomes. Right? We have a lot of knowledge on what they look like. So when Mendel did it, he said, look, I got this purple flower, I got this white flower, I cross them, I take a purple flower and a white one. And by the way he did it is he would take the pollen stem, the stem that had a little pollen on it, the stamen. I'm not going to go into what the structure of a flower is, but he would cut it off with a little pair of scissors, and with tweezers he'd move them over to another flower, and he would literally pollinate, or add the gametes together. In plants, gametes are not sperm or eggs. They're called, pollen is the male gamete for the flower. So the flower is actually a sexual organ for a plant. So parents and white and purple, he mixed them together. Look, you got all purple. So what does that tell you about purple? It's dominant, right? Yeah. Because when you mix the two together, you get mostly one kind. But then he tried something interesting. The next generation, he said, what happens if I mix these two together? They're both purple. All of a sudden I get a white flower. Now that's a question that was asked to you in the dragon lab. In the dragon lab, they asked you that the recessive gene kicked in, right? Yeah. In the dragon lab, they asked you, what if two parents had the same phenotype, but then they have a baby who has a different phenotype? How did that happen? Explain it. Well, the explanation is that there's a recessive gene. And both parents must have been what? Heterozygous, that's right. So these were heterozygous. You ended up with a recessive phenotype. So the law of segregation is during gametic meiosis, the two members of any pair of alleles segregate, unchanged, and are passed into different gametes. So during segregation, so this is his law of segregation, so that each gamete receives only one allele for each of a pair. So what he said was there's somehow these gametes, these four that come out. He did this all with math, by the way. He didn't look at the gametes and try to figure it out. He just said, look, mathematically, this is what happens. That these four particles, and today we know they're gametes. He said these have these things called alleles, and you have two copies, right? Because why are there two? Why are these two sister chromatids connect together? What are they the result of?
Of the crossing over.
No, look at it. How did you get copies of each of these? How'd you get two big A's? No. What happened? This parent only had a big A and a little a. One chromosome with a big A, one chromosome with a little a. Then something happened, and you have two big A's here and two little A's here. What happened? No, they're not. They haven't crossed over here. We're not talking about crossing.
What's it called?
We're not.
It's in my notes. I forgot.
You duplicated it. You replicated it. DNA replication.
That's right.
S phase of the cell cycle, right? So you made a copy. So there they are. Big A, little a, little a. So we know, according to the law of segregation, one of each of these is going to end up in each sperm randomly. So this egg, you have this big A, this egg has little a, right? So that's what the law of segregation states, that they go unchanged, they segregate in the different gametes. Today we know we have crossing over, which is a little bit, it changes things up a little bit, doesn't it? But when the characteristics that he looked at in the pea plant, this is the way it worked out. We worked out very, very clearly because he got very lucky. The genetics of the pea plant were very simple. All right. And notice that this is a diagram that has been simplified, omitting the stage where the second chromatid is produced for each chromosome. So they're skipping the step where we did what? Copied it. S phase is not there, right? That's not part of meiosis. It's not part of mitosis, so that's okay, but it's part of the cell cycle. Then there's this law of independent assortment. By the way, just so you know, we're all clear. I wish we had more time. Sometimes I wish I could teach 8th grade so we could talk about laws. But I'm going to be clear on what a law is. A law is just something that someone notices. It's a pattern in nature. It is not something that people make up. Normally when we talk about laws, they're things that humans make up. Laws in science are things that we notice that happen mostly, like the law of gravity, right, is something we see happen all the time. You throw something up, it falls down, right? So that's a law. There's the law of Newton's laws of motion. Every action has an equal and opposite reaction. We see that. It's a pattern in nature. It's a law, right? Does that make sense? It's not a theory. A theory is still what a theory is an explanation for that law. That's all a theory is. We see this law, now how do we explain that? Does that make sense? Okay. And by the way, theories are very strong. A lot of people think, oh, it's just theory, it's just a theory. It's not just a theory. Theories explain it, and there's a lot of evidence behind theories. Theories don't become laws. You know how some people think that theories somehow graduate to become laws, like a bill in Congress turns into a law. That's not how it works. Theories are theories and laws are laws. Laws are observed. Theories are our explanation of those laws. Okay, law of independent assortment says allele pairs separate independently, so they move to each independent of each other and are passed on to offspring as independent particles, right? That's what Mendel saw when he crossed his peas. What do we know today that makes this not true? all the time. It's true some of them are independent. Like, for instance, this A is on chromosome number 1, let's say. This B is on chromosome number 2. And we know that crossing over kind of messes with this a little bit, but what do we know about this?
That one guy.
That some genes are what? No, dominant recessive doesn't impact this at all. It's still, he still sees that. Linkage. Some genes are linked.
Oh, gene.
So sometimes you get A on one chromatid, and guess what you have on that same chromosome further down? A B. Maybe instead of B being on this chromosome, maybe B's on this chromatid. So if they're linked, in other words, if they're on the same chromosome, then they will not independently assort, will they?
No.
They're going to assort together. So today we're more informed. But remember, Mendel was lucky. He was working with a plant, and he worked with traits that did independently assort. So that helped us figure out, wow, there must be genes on something called a chromosome. Well, today we know that there are many genes on a chromosome, and some of those genes are linked. In other words, they're close together on the same. They're on the same chromosome, so they move together. As you inherit chromosome number 1, you inherit whatever that is on that chromosome, right? All the genes on that chromosome. All right. So those are Mendel's laws of segregation, Mendel's laws of independent assortment. So the idea is that these things are going to assort independently. So you could get big A, little b, big A, little b, little a, big B, and little a, big B. Today we know we have crossing over that can make 4 different ones instead of just 2. You see that there's just 2 options here, right? Today we know that we can have crossing over if they're on the same chromosome. These are on 2 different chromosomes, so these are always going to be, either you're going to have these options as far as on different chromosomes. But if they're on the same chromosome, then you can have crossing over, and that'll rearrange that chromosome and give you 4 different chromosomes instead of having just 2 different chromosomes. Does that make sense to everybody? All right. Epistasis. Epistasis is a little. It's a big word, fancy word. It sounds like to people, but it's actually something that makes absolute sense if you think about it. So you have genes. Today, by now, let's go over. We've gone over transcription and translation. We've gone over how DNA is replicated, semi-conservative model. We've talked about the idea of transcription. You have to make mRNA. mRNA gets turned into protein in the cytoplasm, right, as translation. That protein, they make enzymes. Here's an enzyme, tyranase. So what tyranase does is it takes tyrosine, which is it's a dark pigment, right? And it takes this thing called tyrosine, and this tyranase makes melanin. So this is what makes the dark pigment. So this mouse is albino if it doesn't have tyrosinase. If it doesn't have this enzyme, then it can't make the pigment melanin. That's what's going to make the albino, because it can't make this thing. This melanin is the pigment that makes that mouse dark. Well, it turns out that there's another gene, gene B, that makes this TYRP1 that encodes, gene B encodes for tyranase-related protein, TYRP1, which determines the level of pigment expression. So this one makes even a lot of pigment expression. Remember, is how much is transcribed, how much is translated. If it's not transcribed, it doesn't matter that you have the gene, you're not going to see it. Does that make sense? So if there's a lot of this, then what's going to happen is, in other words, if you have one version of TYRP1 and it's working, then you're going to get a lot of melanin showing up, and you get a really dark mouse. If you have not a lot of this, then you're going to get a brown mouse, because you're going to have melanin, because this one tyrosinase made melanin, right? But how much melanin are you making? Well, that's controlled by gene B. Does that make sense? So what you have is one gene, gene B, influencing gene C. So one gene influences the other gene. We call this epistasis. So when one gene impacts the other gene, we call it epistasis. So it's very, very clear that this is an issue in a lot of different situations. And there's some problems there. And you have this one, don't you, as a homework assignment? So I'm going to go ahead and let you work on what the answers to these questions are, right? So if you have this phenotype, in other words, a recessive B and a dominant C, you have brown. If you have dominant B and recessive C, then you have an albino. Even though, it's interesting, even though you're making a dominant B, so what you have recessive C, which means you're not making any melanin. That's why it's albino, right? Where even though you have the B gene that turns on that gene a great deal, the protein it makes, the protein that this lowercase c makes, is not melanin. It's something that's broken. It doesn't work as a pigment. Whatever it's making, it doesn't work as a pigment. Right? Does that make sense? So that's why you could have a big dominant B and recessive C's and nothing happens. Here you have a dominant C, which means you're making a really good melanin. It's actually a great pigment, but because you have the recessive B, it's not a lot. You're not making a lot of it, so you get a brown mouse. Now, if you have the dominant form of the B, whether it be big B, or big B, little b, that means you're making a lot of it. And you have the dominant form of the C, which means you make melanin, the good copy of the melanin, the strong pigment. Then you're going to have a black mouse. Does that make sense? So that's why we do a heterozygous chart. Because there's three different phenotypes depending on your genotype. Does that make sense?
Yeah.
And that's what's going on with your dragons. In your dragons, you have one set of genes that are incomplete dominance, right? That even though you have big T, little t, now incomplete dominance is not quite epistasis, right? Incomplete dominance is a little bit different, right? That's when you have only one gene. You're only talking about one gene. Epistasis, remember, you're talking about one gene impacting the other gene. Is that clear? In incomplete dominance, you have one gene. The three different genotypes give you three different phenotypes. Slightly different, okay? All right, so that helped? Understood? So it should be okay. I think you can do it. Now here's an example of a Labrador. A Labrador, this is a really interesting problem. You've seen yellow labs at all? Something looks like this? You can have little e, little b, little b. Same thing as the mouse, right? If you have it, the capital E, then you produce a form of melanocyte that stimulates the receptor. And if you have gene B, it encodes for a level of pigmentation. So this makes the melanin. This one encodes how dark it is. So if you have the recessive type, the way you see here, little e, then you have a yellow lab. In other words, there's not a lot of pigment in the dog's fur. But if you have the dominant B, then you could either have a chocolate lab if you have the recessive Bs, right? So it's not very dark. Or you can have a black lab because you have a B that is, has a capital, so you're making a lot of it. So you either have the dominant form of B or you have the recessive form of B. And if you have the recessive form of E, the way you see here, then it's not going to make any of these pigments in the fur, so you end up with a yellow lab. So it's exactly like the brown and black mouse, right? And white albino mouse. Yeah.
Would the dog be albino too, or just?
It's not albino because it's a little different. So
I thought it was the same thing.
You're right that it's the same pattern that we saw with the mouse, but it turns out that the mouse's genetics are a little simpler. The dog has other pigments, other genes that make melanin in different ways. This particular gene controls the fur color. But the dog has other pigments, other cells that are making other pigments, so it's not quite an albino, even though it's lighter.
So this control the fur color. This basically determines what color it's going to turn out.
How dark, right. So this makes the melanin a receptor. Look, read it carefully. It says, big A produces a form of melanocyte-stimulating hormone receptor that is extremely active and constantly signals the production of eumelanin. The recessive allele does not signal signal as energetically. So it's not producing as much, but it's still producing some. Where with the mouse, the example we saw earlier, there was the same idea, right, of epistasis. But with the mouse, the recessive form produced no melanin. And so that was easier to understand because either you're making it or you're not. This one's, the lower case is making a little, but not a lot. Does that make sense? So this is why this is controlling this form. Now, interestingly, this because it's little b, it's producing a little bit of melanin, and it's it's not used producing a lot of it because this is controlling how much, right? The level of pigmentation. This dog has a yellow lab with brown nose and brown eyes and brown lips. But you could have a yellow lab. Why do you have a yellow lab? Because there's a third gene, gene C, that alters the color of the yellow Labrador. The homozygous dominant dog will appear dark yellow due to the large amounts of I can't pronounce that, but phenomelanin, and heterozygous and homozygous recessive dogs being progressively lighter. So now there's even a third shade with a third gene. So before, remember we talked about 2 genes with the mouse. Now we're looking at 3 genes, E, B, and C. And they're all impact. So that's why we get 3 different colors. Now, can you imagine with humans why we would have so many different shades of skin color? Like we looked at the skin color activity, right? That we call that scale, right? From 0 to I think it was 5.
Yeah.
We have 3 genes with 3 different alleles, and actually I think it's up to, like I said, 6 or 8 genes now. 6 or 8 genes with so many different alleles, each one having an impact, like very much like in this example with dogs or with the mouse, is exactly what we're dealing with. So whether we have all these different shades, and so it's very difficult to say these 2 are going to be this way or those 2 are going to be that way.
Does it have the same thing in humans that it has?
Is it epistasis the way it is in humans? Is skin color an epistatically controlled phenotype the way it is in dog color in dogs or rat mice? I'm not sure about that. I don't know. It certainly could be. Certainly could be. So we could take a look at this, and again, you can answer all these questions in genotype and phenotype. And it's just a dihybrid cross. Here you're dealing with 2, E and B. And of course, we could add the third one if you really wanted to, add A, B, and C and do a trihybrid cross the way we did with skin color, right? But they're asking you just to do a dihybrid cross, and I think you can figure out what colors you have 3 colors, just like you did with the mouse, depending on whether you have little e, big E, little e, or big B, etc. Okay? Not that hard. I think it's pretty straightforward. Then with this cat one, inheritance of domestic cats, here you have all the different kinds of inheritance that you can get, right, that are in the packet. The definitions are there, epistasis, multiple alleles, which is what we talked about. And I'm going to say no with skin color in humans because they gave us an example of multiple alleles, right? And it's not an example of epistasis. So, but I'm not sure if there is a form of epistasis going on in human skin color is my problem with answering that definitively for you. Recessive dominance, incomplete dominance, lethal genes, pleiotropy, sex linkage, environmental effects, all these, right, help influence the cat color. So read these carefully. We've covered almost all of them. I think the only one we didn't talk about was pleiotropy, which I'll try to explain right now. But then you'll see here that you have these wild forms, these mutant forms. You have the phenotype, all right? And you have, so if you have big A, you get this color. If you have little a, you get this color, etc. I know some of you are allergic to cats. I know some of you don't have any cats. You're not going to go out to your cousin's house to go find some cats. So just go online, find some pictures of cats randomly, and do the lab. The lab is there for you to collect the data. It's really easy to do. So let's talk a little bit about pleiotropy. It says the white gene, big W, also affects eye color and can cause congenital deafness, 1 gene with 3 effects. So with epistasis, we saw one characteristic, the color of the mouse or the color of the dog, right? Being influenced by one gene influencing the other gene, how well it expresses, right? With multi-genes or polygene, right, multiple alleles, we saw in multiple alleles were the ABO blood group, right? You have 1 gene, but 3 alleles, either A, B, or O, right? And that gave you different kinds of phenotypes. And then you had, polygenes aren't here, but we talked about polygenes, right, with skin color. So you have 3 genes impacting skin color. But now it turns out that you can have 1 gene controlling 3 different outcomes. So you can have, so let's go over and think about this. With skin color in humans, you have many genes. Let's call them 1, 2, and 3. All 3 genes influence what? Skin color. Right? That was polygene. With multiple alleles, what you have is 1 gene, and let's call it, let's just call it blood type. All right? The blood type gene has what? 3 different alleles. A, B, and O. And I should write I. Right? Remember that? That's how we write that. And these give you 3 different phenotypes, right? Or actually 4 different phenotypes, because you can have, if you have 2 AAs, you have A. If it's AA or AO, then it's A. If it's B or BO, it's B. If it's OO, it's O. But if it's AB, it's AB. So you have 4 different phenotypes coming from these polygenes. We good with that? So that's 4 different phenotypes with 1 gene, but 3 alleles. Here's 3 genes with 1 phenotype. Then you have let me shrink this down a little bit. And then you have the idea of epistasis, right? And that's when you have 1 gene, gene A, impacting gene B, right? Regulating gene A. The product of gene A influences gene B. So you get different phenotypes, right? So that's epistasis, and we just covered 2 examples, the dog, the Labrador and the mouse. Then this last one is pleiotropy. And pleiotropy is just simply you have, it's not simple, but you have 1 gene, let's call it gene 1. And because you have gene 1, you have 1 phenotype, phenotype 1. But also that same gene, the same allele, no other genes involved, no other alleles are involved, also gives you a different phenotype here. Also impacts a different, I shouldn't even say phenotype, so that I should change that. It impacts 3 traits. So 1 gene can impact 3 traits. So 3 different traits. And the example they give here is, so you get the white gene W also affects eye color. So the cat can be white, also it changes the color of the eye, but also makes the cat deaf. So 1 gene can make it white. Let's say, I don't know what color, but let's say blue eyes. And it's deaf. All from 1 gene. 1 gene product, 1 allele. We call that pleiotropy. That happens in life as well. These are all non-Mendelian. These don't follow the Mendels Laws because, again, Mendel was dealing with peas he got very lucky. The genetics were simple. They were either dominant or recessive. But we know today that we have polygene, epistasis, multiple alleles, pleiotropy, and now, of course, don't forget incomplete dominance. Oops. And we have, what's the other one? Codominance. Very good. So all these are different ways of inheritance that impact the outcome. They can impact the outcome. Is that good? Everybody get it? And then here's some of the they give you, okay, this is what non-ability looks like, this is Siamese looks like, etc. And the last, this is your data sheet. So you have to go collect the data online. Go and find these. It's easy to find. Just go look up cats. They're everywhere on the internet. color pictures, choose, just as soon as you see, is this cat a goatee color, is it black, is it, et cetera, okay? All right, that's it. Have a good one. Wow, I was able to do that in one period. Did this help, by the way?